So hello everyone, welcome back to Hasvora Education. Today we will be solving Biology 9700 paper 34 May-June 2025 session. This is the predicted paper that was published on the site of the CrackAlevel. So some of you were requesting me to solve it as soon as possible. So here I am, but this worksheet only contains the question ones that are related to the dilutions. But if you want a question 2, I have already published a video on what I expect to come in the paper, and it also has a question 2, so go watch that video if you haven't till now. So now let's begin. The first question says, vegetables such as carrots contain sugar. Potassium permanganate solution can be used to identify the presence of the sugar. So most of the sugars are actually reducing sugar, so they can carry out a reaction known as reduction and can reduce the oxidizing agent potassium permanganate. So we would probably be identifying this trend in this question. So the sugars change the color of the potassium permanganate solution from purple to colorless. You will measure the time taken for potassium permanganate solution to turn colorless. With sugar solution of known concentration, you will use the result to estimate the concentration of sugar in a carrot extract. You are provided with the material shown in table 1.1. So S is 1 mole per dm cube sugar solution and it has non-hazard. W is the distilled water. A is the sulfuric acid. It is a harmful irritant, so it would have a high risk. K is the potassium permanganate and it is irritant, so it would have a medium risk. C is carrot extract. It has non-hazard. If any solution comes into contact with your skin, wash off immediately with the cold water. It is recommended that you wear suitable eye protection and wear gloves to protect your hands when using A and K. You will need to prepare different concentration of sugar solution. Record time taken for the Ka to become colorless for each of the different concentration of the sugar solution and for the carrot extract C. Use a result to estimate the concentration of the sugars in the carrot extract C. You will need to use proportional dilution. So as I told you earlier that proportional dilution is the same as simple dilution. Okay, so it is the same as simple dilution. To make 5 different concentration of the sugar solution, you will need to prepare 10 cm cube of each concentration. So we are going to be preparing 10 cm cube using S and W. Table 1.2 show 2 of the concentration of the sugar solution you will be using and how to prepare them. Decide which 3 other concentration of the sugar solution you will use. So they have given us 2 of the solutions from the 5. So we have to just make 3 other solutions other than that. So A part 1 says the table 1.2 to show you how to prepare the other concentration of sugar solution you will use. So if they have given us 1 and the 0.0, so let's predict at what concentration gap we will maintain to have 3 concentration between these ranges. So if we keep, for example, 0.2 gap, so it will be 0.0, 0.2, 0.4, 0.6, 0.8 and 1. So it would be actually 6 concentration, and we have to have 3 concentration between the range, while this will the 0.2 gap will actually give us 4 concentration. So in order to maintain the 3 concentrations, let's check about 0.25. So with 0.25 we can have 3 concentration between the range. So the first one will be 0.25, then we will have 0.50, and then the other one will be 0.75. So this will be the 3 concentrations. Now we will use the M1V1 and is equals to M2V2 to find that what volume are we going to be picking and how are we going to be preparing it. So M1V1 is equals to M2V2. M1 is the stock solution, the 1%. We do not know what is V1 and what volume we are going to be taking out. M2, so we are going to be preparing the 0.75 first, so let's take it 0.75, and V2 we are going to be preparing 10 cm cube of each solution. So V1 comes at as 7.5. Now let's do it for the 0.50 as well. So it will come as V1 is equals to 0.50 into 10, so it will be 5 cm cube. And similarly, the V1 for the 0.25 will come as 2.5 cm cube. So this actually means that we are going to be taking out this volume from the S. Okay, so this will be 7.5, this will be 5.0, and this will be 2.5, and the rest we are going to be adding the water to make the 10 cm cube mark. Okay, so it would be 2.5, then 5.0, and then 7.5. Now let's make the lines. Now let's make the lines. Okay, just make sure that the the gap between each of them is the same. So this will come as like this. Now let's draw another line. Now let's drag it like here, and this will be about here, and the other end will be like this. Let's copy it again, and this will come a bit lower like this. Okay, so this is how we are going to be preparing our table. So we have made the table. Okay, so we have made a table. Now let's move on. Carry out step 1 to 11. In the beakers provided, prepare the concentration of the sugar solution as shown in table 1.2. Label the test tubes with the concentration of the sugar solution prepared in step 1. So what are we going to be doing? We are going to be first marking the test tubes after the preparing the solutions in the beakers, and then we are going to be then put 1 centimeter cube of 1 mole dm cube sugar solution into the appropriately labeled test tube. Repeat step 3 with other concentration of the sugar solution. So we are going to be putting 1 cm cube of the other concentrations into their respective uh, test tubes. Put 1 cm cube of the A into the each of the test tubes. So A is the sulfuric acid. Just let me write it here. Shake gently to mix. The reaction will start as soon as you put K into test tube, which is step 6. Keep the timer running continuously until the end of the step 7. So we are going to be starting the timer as we add the first the first you can say as we are going to be starting the reaction for all at the same time. Actually, when we add the first cm cube of the K into the any of the test tube, we are going to be starting the timer, and then we are going to be just you can say lapsing each time as the endpoint is reached for each of the concentrations. So measure the time taken for each concentration to reach the endpoint. At each endpoint is reached, record time taken in A part 2. If the endpoint has not been reached after the 600 seconds, record the time as more than 600. Record your results in the appropriate table. So we are going to be making another table here. Let's make it now here. So this is going to be our table. Then we are going to be drawing a line here, and then we're going to be the heading section. Then just let me pick up the ruler, the third row, and then the fourth and the fifth one. So we are going to be making our table like this. In the first column, it will come the concentration of S solution. I am not writing the full form as I don't have enough space. And then mole dm cube. Then time taken for color change. Okay. Now let's write the concentration in the ascending order. The first one was 0.0, then 0.25, then 0.50, then 0.75, and then 1.0. So let's just assume, as we are not performing the experiment, let's assume that uh, and for sure it will be, the trend will be like this, that as the concentration is decreasing, it will take more time to decolorize. So let's take the first value as more than 600. Okay, be more than 600. Then let's take this one as 587 seconds, this one as 570, not 570, let 5, just you can say 542 seconds, then 498 seconds, and then this one will be about 421 seconds. Label a test tube C and put 1 cm cube of C into a test tube. So C is actually the unknown extract that we are going to be predicting using our dilutions. Put 1 cm cube of the A into test tube and shake gently to mix. Put 1 cm cube of K into test tube and start timing. Shake gently to mix. Measure the time taken for it to reach the end point. Record time taken in A part 3. So let's just assume its time to be about 456 seconds. Okay. And one tip I want to give you here is that you are going to be always and always writing your time in the whole numbers. In even if it comes, and for sure it will come in decimal as you are going to be using a stopwatch, but you have to round it off to whole numbers. Estimate the concentration of sugars in C. Now let's have a look into our table. So the 458 second will be about something between this. Okay, it will be something about between this. So so what we could do is that um, we can just predict any concentration between these two these two marks, like between the 0.75 or between 1.0. So let's take it as it is closer to the 421 mark, so let's just take it as 8.2. Now suggest how the procedure could be modified to improve the accuracy of your estimate in A part 4. So this is actually the plan investigation, the question, and in which we are actually giving them the suggestions that how this experiment could be improved. Like for example, in this we are estimating the concentration of the C, but how are we going to be uh, improving and modifying this experiment so that we could have more precise concentration and we could estimate more precisely the concentration of C. So the first one can be repeat the experiment with each concentration 4 to 5 times and calculate average time or mean time. The other improvement could be you could also, what you could do is that uh, time each of them separately. Okay, so let me just adjust the pen. So calculate So calculate or time for each concentration separately. Or the other thing that you could do is that what else can be done is that you could use a colorimeter to determine the determine endpoint of experiment. So these are the three improvements that can be made to modify the experiment. Part 4 says part 6 says the student used the same procedure to compare the concentration of the sugars in three vegetables, carrot, potato, and onion. State one variable that needs to be standardized. So the volume of the sample should be kept the same. Volume of sample should be kept the same. Suggest how the student could extend their investigation to estimate the concentration of the starch in vegetable extract. So this is now a new, you can say, experiment, and now we are going to be providing them how a brief detail of how the experiment should be conducted. So what would we could say that take 5 different concentration of starch solution prepared using simple dilution is more convenient, okay, because it gives you small intervals, as wider intervals are less convenient. So using simple dilution, and also you can provide the numerical values of the concentration that could be used: 1%, then the 0.8%, then 0.6%, and 0.4%, and 0.2%, and you could also write the percentage at the end of all these values one time. Then what else can be done? Cal- Calculate the time it takes for iodine solution to show color change. So this is how the starch could be actually estimated. The concentration of the starch in a vegetable extract could be investigated. So this was the part of the question. B part says the banana produce ethylene gas which causes them to ripen. The production of ethylene gas continues after bananas are removed from the plant. The ethylene gas decreases the post-harvest life of the bananas. The post-harvest life is the time after the harvesting when bananas are suitable to eat. Potassium permanganate can be used to increase the post-harvest life of the bananas by oxidizing the ethylene gas to form water and carbon dioxide. So ethylene gas is actually decreasing the post-harvest life. Why? What is potassium permanganate doing? It is converting that ethylene gas to water and CO2, and for sure it will increase the post-harvest life. So an experiment was conducted to determine the effect of different quantities of potassium permanganate on post-harvest life of bananas. So the results are shown. So we have a range of the mass of potassium permanganate on the x-axis from 0 to 8, and on the y-axis we have the range from the lowest is 9.3 and the highest is 13.65. So we will just assume 14 as a reasonable value to have a suitable scale. So on the x-axis we have 1, 2, 3, and 4 boxes, full boxes. So we could use 2 as a suitable scale: 2, 4, 6, and 8. It was the mass of the potassium permanganate, mass of potassium 7 magnate, and the unit was grams. On the y-axis we have 1, 2, 3, and then 4 and 5. Okay, this will be 4 and this will be the 5. So 14, let's divide 13.65 by, which was the highest value, divided by 5. So it comes as 2.73. Let's take 3 as a reasonable scale: 3, 6, 9, and 12 and 15. What was the unit? Let's check it from the table. It was mean post harvest life in days. So mean post harvest life, and it was in days. Now let's plot the values. Okay, the first reading is 09.30, so 0 will be here. Okay, now we have over one big box on the y-axis equal to 3. So what will the smallest box, like this small box, equal to? It will be equal to 3 divided by 10, which will be 0.3. So I have already showed you the trick. So 9, what we'll be doing to plot the reading, 9.3 minus 9 divided by zero point 0.3, it comes as 1 unit. So it would be somehow about this point, 0 and 9.30. Let's draw a circle around it. Now what will be the next value? It would be 2 and 11.25. 2 and 11.25. Let's do the same. 11.25 minus 9 divided by zero point 0.3, it comes as 7.5 units. So 2 and 7.5, 2 and 7.5, this is the plot. Now let's move 4 and 12.6. So 12.6 will be actually 2 units above from 12, so it will be this point. Then above the sixth is 6, 13.65. So 6 and 13.65, 13.65 minus 12 divided by zero point 0.3, it comes as 5.5. So this will be the point, and after that the last coordinate is 8 and 11.45. So 8 and 11.45, 8 and 11.45. So 11.45 minus 9 divided by zero point 0.3 comes as 8 units. So this will be the coordinate. Okay, so now let's join them with straight line. This will be the one straight line from here to here, then another straight line from here to here, and then another straight line, and then the last straight line. This will be the graph of this question. Use your graph in figure 1.1 to predict the post-harvest life of bananas if the mass of the potassium permanganate is 6.8 grams. So show your graph to how you obtain the answer. So what are we going to be doing is that we are going to be just interpolating our graph. What does interpolating means? That we are going to be marking this point. For example, this is 6.8. Okay, this will be 6.8. Let's just interpolate it by using dotted lines. This is how we are going to be using the dotted lines. So it will come as this point, about this point. Okay, so this will come as about 12.65 days. So 12.65 days. Now suggest why the post-harvest life of the bananas decreases when more than 6 grams of potassium permanganate is used. So now let's have a look at what is the role of potassium permanganate here. So it is actually oxidizing ethylene to CO2 plus water. So now what can CO2 cause, or what can water cause? What water can actually disrupt the osmotic balance of the plant, and CO2, it could combine with that water to form carbonic acid, and it could just interfere with the pH of the plant. So we could write about the pH. So CO2 or carbon dioxide combine with water to disrupt pH of the plant. So you could give any reason from these two. So let's move on to the next question. This is the marking scheme. You can have a look at it. The question number 2 says plant extract contain molecules which have many uses in the industry, and as medicines, it is important for scientists to be able to estimate the concentration of these molecules in plant extracts. You will estimate the concentration of molecule R in the sample of plant extract U. So you will be actually the unknown concentration that we are going to be just determining at the end. So you are provided with the materials shown in figure table 1.1. So 10R is a 10% solution of molecule R. Then the W is distilled water and U is the unknown concentration of the molecule R in the plant extract. A is the sulfuric acid. K is the potassium permanganate. If any of the solution come in contact with your skin, wash off immediately under cold water. It is recommended that you wear suitable eye protection and wear gloves to protect your hand using A and K. You will need to carry out a seal dilution. So now we are performing the seal dilution actually. In this part we are performing the seal dilution, okay, and they are going to be always stating a condition together with the seal dilution, that whether you are going to be halving the concentration, and this is what we actually do most of the times, but sometimes they actually ask us to uh, just divide it by 10, the 1/10 of the concentration after each successive dilution. So in that case, if for example we have an original concentration of 1%, it will become 0.1, then 0.01, then 0.001, and like that. But here they are actually asking us to reduce the concentration by half between each successive dilution. You will need to prepare 4 concentrations of the molecule R solution in addition to the 10% molecule R solution, which is the 10R. So after the serial dilution is complete, you will need to have 10 cm cube of each concentration available to use. So complete figure 1.1 to show how you will prepare your seal dilution. Figure 1.1 shows the first two beakers you will use. To make your seal dilution, you will need to draw 3 additional beakers. For each additional beaker, add labeled arrows to show the volume of molecule R transferred and the volume of distilled water added. So they have given us 2 beakers, we are going to be making 3 others. So now you can see we have 20 cm cube of 10% R in the start, but we are remaining with 10 cm cube of 10% R solution to use, which means that 10 cm cube of 10% R solution is actually transferred, okay, it is transferred. And now we are also going to be making an a straight arrow like this. Let me redraw this arrow. So this will be the arrow, and we are going to be adding 10 cm cube of W. Then we are going to be making another arrow, and we are also going to be adding this to another beaker, and we are actually making 20 cm cube of it, but we are transferring 10 cm cube of it to the other beaker, so it actually remains 10 cm cube in the end. So it will be 10 cm cube of 5% R solution to use, okay. Then we have transferred this will be another beaker, we are going to be drawing another beaker. Then we will have another arrow, this will be another arrow, we are going to be transferring 10 cm cube of W again, and this will be 10 cm cube of 5%, actually 5% are transferred. Now we are going to be making another beaker in the same way. This will become 10 cm cube of W, and then this will become 10 cm cube of 2.5% transferred, 2.5% R. This will become the, actually I did not have labeled this first one, so this will be 10 cm cube of 2.5% R solution to use, and this will become 10 cm cube of 1.25% R solution to use. Then we are going to be making another and the last beaker, which is going to be this one, and we are also going to be adding 10 cm cube of water to this one, and 10 cm cube of 1.25% are transferred. This is going to be the last beaker, and we will have 10 cm cube of 0.625% R solution to use. But this will actually be 20 cm cube in the end, because we are not actually transferring it from it anymore. So molecule R changes acidified potassium permanganate solution K from pink to colorless. The lower the concentration of molecule R, the greater the volume of R needed to change the potassium permanganate from pink to colorless. So they have actually told you the trend, that the lower the concentration of the molecule R, the greater the volume is actually required. So they've actually told you this trend here in this line. Just let me highlight it. So they have already told you in this line. Okay, so the lower the concentration, the greater the volume. Now carry out step 1 to 12. Prepare the concentration of solution R as decided in A1 in the beakers provided. Label 5 test tube with concentration of R you prepared in step 1. Put 1 cm cube of A into test tube labeled 10%. Put 1 cm cube of K into the same test tube and mix well. Put the nozzle of the clean 10 cm cube syringe into the beaker containing 10% R. Pull the plunger out of the 5 cm mark so the 5 cm cube of 10% R syringe enters the syringe. Put the syringe contain 10% R into test tube from step 4. So what are we actually doing is that we are just uh, trying with each of the concentration that we have prepared, that how much of the drops are actually required to decolorize the KMNO4. So you will be counting the number of drops needed to change K from pink to colorless. Gently press the plunger to add 1 drop solution containing R as the mixture stays pink. Repeat step 8 and 9. Count the number of drops released to reach the endpoint. If the number of drops needed to reach the endpoint is more than 100, record more than 100 and record the results in a part 2. Repeat 3 to step 11 for each of the concentration of molecule R prepared in step 1. Now let's make a table for the number of drops and the concentration. So this will be the table. This will be the first row for the heading, the concentration of solution. What was the solution? Name of the solution was R. Okay, so concentration of R was in percent. Okay, and then number of drops needed, or you can also write number of drops needed for color change. This is going to be the first one, then the second one, then the third one, and then the fourth and fifth one. Let's write it in the ascending order. 0.625, then 0 not 0, it was 1.25 actually. So 1.25, then 2.5, then 5.0, and then 10.0. Let's assume that the 0.625 took the most number of drops as it has the lowest number of concentration, so more than 100. So 1.25, let's assume it take 92. 2.45 took actually 81. As it is sealed dilution, so it will be actually greater gap between the values. So let's take it as 70. Let's take it as, you can say, about 64. Okay, and the last one will be about 38. State two significant sources of error when measuring the dependent variable. So what was the dependent variable? It is the number of drops, which is actually depending on the independent variable, which was the concentration. So the significant source of error when measuring the drops is that unequal volume, or you can say more than one drop can be added, or you can also say that uh, unequal drops of solution are added, or the other can be difficult to determine the endpoint. The endpoint is actually when it becomes colorless. So this will be the other error. Suggest how you could improve the procedure to reduce one of the errors. So for the color, we could use a colorimeter, and for the drops, what we could do is that measure volume instead of number of drops. So you have to list only one of the improvements. Repeat step 3 to 10 with the unknown concentration of molecule R, which is in the U. Record your result in A part 5. State a number of drops of U needed to reach the endpoint. So let's take it as, just take it as 48. Okay, it has no units. So complete figure 1.3 to show the position of each of the percentage concentration of molecule R solution recording A part 2. So it was actually 10%, then 5.0%, then the 2.5%, then 1.25%, and then it will become a little bit clogged up here, so 0.625%. Now they are telling us use the result in A part 2 and part 5 to estimate the concentration of molecule R in solution U. So what are we going to be doing is that solution U actually takes drops of uh, solution R 48. So 48 will be essentially between these two values. So let's take it between 5 and 10, as it is more closer to 38 or a 10% solution. So we will take it as about 9%. Okay, so this will be about 7.5 actually, but we do not have to mark. I'm just showing to you that how we are going to be performing. So the 9 will be somehow about here, 9.0% U. So it will be actually U, but you are just gonna write here U, the letter U, not 9%. Okay, so show this estimate of concentration of U in on the figure 1.3 by placing the letter U on the line in correct position. So we have already done this. B part says a student investigated the concentration of molecule R in different extract of the plants. The student added a fixed volume, so they are keeping the volume fixed of the extract to acidified potassium magnate, and the time how long it takes to reach the endpoint. So the results are shown. So you can see that in the first column there is actually not any numerical values but letters. So it means we are going to be drawing a bar graph. I have also provided this guide in the revision, or you can say the expected paper, which I posted yesterday. So you could just go if you can and watch it if you do not know that in which case we are going to be making a bar graph or a line graph. Other than that, the examiner also tells us to draw a bar graph. So they are saying plot a bar chart of the data shown in figure in table 1.2 on the grid on figure 1.4. So we are going to decide that how we are going to be distributing over x-axis in a way that there is equal distance between the bars, and also the bars are of uniform width. So for example, if we keep it as 5 of the bar width is actually of 5 small boxes, then we could essentially make here 5 bars here. So let's keep it here as 5 small boxes. On the y-axis we are going to be plotting the time. So the highest time is 59 seconds. Let's see how much boxes we have on the y-axis. So 1, 2, 3, 4, 5 and 6. Let's keep 10 a reasonable estimate. 10, 20, 30, 40, 50 and 60. So let's check the first value, which is B. It is at 15 seconds. So 15 will be at the midway between the 20 and 10. So it will be here. Now let's draw the bar. This will be B. Now let's check for C. C is 59 seconds, one small box below the 60th mark. So it will be about here, okay? So this will be C. Then let's check for D. D is 42. So 42 will be somehow about here. This will be D. Let's check about E and F. So E is 10 and F is 7. This is 10 and this is other will be 7. This one will be 7. So this will be E and F. This is how you will be drawing your bar chart. So suggest which plant extract would contain the highest concentration of R. So as we know that the higher the volume needed, it means the lowest the concentration is present. So most of you, the mistake will be done here is that they will just look into the graph, they will see that the highest time is of the C, and they will just write their plant C. So but here the point is actually that the lowest time taken actually ensures the greatest concentration, and they are actually asking us about the greatest concentration. So the lowest time taken is by plant F. So plant F. Scientists have suggested that a different molecule in a plant extract might act as an antibiotic. So antibiotic is actually used against the bacteria. The scientists tested this by spreading bacteria over the surface of the agar gel containing the nutrients. So actually the bacteria would grow on that nutrients, putting small drops 3 micrometer cube of different concentration of the plant extract into the separate wells in the agar gel, incubating the agar gel for 20 hours and measuring the inhibition area. So where the actually bacteria was not observed and it was actually removed by the plant extract molecule. So this was done for the each of the concentration. So we could see that inhibition area is increasing as the concentration of solution of molecule R is increasing. So use the graph in figure 1.5 to estimate the concentration of the plant extract that results in inhibition area of 92 millimeter cube. So 92 millimeter cube will be somehow about, it would be somehow about between the mid of 180. This will be 90 and this will be 92. Okay, not 92, the first box will actually be 92, this one. Okay, so let's interpolate this graph. So this is where it will actually end up, and let's check the concentration. So it will be the first box after the 20th. So as it is it has one small box is equal to 2 here, so it will be actually 22 microgram per cm cube. 22 microgram cm cube. suggests how molecule in the plant had may inhibit the growth of bacteria. So for growth, there the protein is actually required. So it would actually be inhibiting the protein synthesis via translation and transcription. So what we could write is that it can it would inhibit the enzymes that are being uh, used in the protein synthesis, or you could say that it is actually inhibiting the translation and transcription. So we could write that the plant extract inhibits the enzymes involved in protein synthesis via translation and transcription. Do not use the abbreviation, use full. You will write full and okay and transcription. Also, you could write that it releases chemicals that destroys cell wall and cell membrane of bacterial cell. So this is what you actually could write for 2 marks here, 2 points. Other point that could be is that it could have a small, it could have a more water potential and less solute. So what would actually happen that the water would move by osmosis to the bacterial cell, and because of trigger pressure it could uh, burst and cell lysis could occur. So any point can be valid unless and until it is related to the inhibition. So now we are going to be performing this last question of this paper. So energy drinks contain a high concentration of carbohydrate such as the glucose as a source of energy. Glucose is a monosaccharide which can be quickly metabolized by the body cell releasing energy during the exercise. Glucose changes the color of potassium permanganate solution P from purple to colorless. The endpoint is when P is completely colorless. You will need to perform the different concentration of glucose solution, record time taken to reach the endpoint for known glucose concentration, and use your result to estimate the concentration of glucose in an energy drink U. So we have been provided with 20% glucose solution G, W is the water, A is dilute sulfuric acid, and P is the potassium permanganate solution, which is a low risk irritant. So U is the energy drink, and it is recommended that you wear suitable eye protection. If A or P come in contact with your skin, wash off immediately under cold water. So let's move on. You will need to prepare different concentration of the glucose solution. The lowest concentration you will prepare is 12%, and you will need to prepare 10 cm cube of each concentration using G and W. Table 1.2 shows how to make up two of the concentration of the glucose solution you will use and decide which other concentration of solution you will use. So we are going to be performing, actually we are going to be preparing the concentration between these two ranges. So let's take a gap of 2, and it will become actually 18, then 16, and then 14 actually. So let me just make it a little bit in proportion. So it will be 18, this will become 16, and this will become 14. Let's use m1v1. So m1v1 is actually equal to m2v2. So the stock solution is 20, we do not know v1, m2 is 18 in this case, and 10 cm cube of each. So it will become 180 divided by 20. 180 divided by 20 becomes v1 is equals to 9. So and for the others it will become v1 is equals to 160 divided by 20 is equals to 8, and then for the others it will become V1 is equals to 140 divided by 20. Okay, so it will become 7 cm cube. So now let's write it into the table. This will become 9, this will become 8, become 7. So the rest will be the water. Okay, so it will be 1, it will be 2 here, and it will be 3 here. Now we are going to be just drawing the lines here like this. Just ensure they are in proportion. I do not have a ruler right now, so I'm just drawing freehand. Let's move on. Carry out step 1 to step 8. Prepare concentration of the glucose solution as shown in table 1.2 in the beakers provided. Label test tube where the concentration of glucose prepared and put 1 cm cube of 20% glucose solution in the appropriately labeled test tube. Repeat step 3 with each of the other concentrations of the glucose and put 1 cm cube of sulfuric acid, which is the A, to the test tube. Let me write down the sulfuric acid here. So sulfuric acid. Shake gently to mix. The reaction will start soon as you put P into test tube. Do not stop the timer. Keep it running continuously. So we are going to be starting for each for all the reaction at the same time. Put 1 cm cube of P into each of the test tube and start timing. Shake gently to mix. Measure time taken for each test tube to reach the end point, which will be purple to colorless. As each endpoint is reached, record time taken in the space provided for the raw results. Do not stop the timer. So as we are starting for each of the experiment at the same time, we are just going to be lapsing. For example, we are going to be starting 0.00. For example, if the highest concentration is going to be showing us the time, it will take us actually less time. So for example, if it shows up at, like, for example, 124 second, which will be about some 2 minutes and something some seconds. So if it shows up at 124 seconds, we are not going to be stopping the timer. We are just going to be noting the time in this raw results, and we will let it run till the end. Okay, so this is why we are going to be recording here in this space for raw results. Now let's move on. Record your results in the appropriate table. So now we are going to be making a table here. This is going to be a table, and now this is going to be the two of the columns. The first row is for the heading, and which is the concentration of, what was the concentration of G actually, and it was in percentage. Then time taken for color change. You are also gonna write color change. I'm just writing a short form because I do not have enough space. So the unit will be S. This is going to be the first, then the second, then the third, then the fourth, and the fifth one. Okay, we are going to be starting with the lowest concentration in ascending order. So this will become 12, 14, 16, 18, and 20. Let's suppose the smallest concentration takes more than 600. So more than 600. Let this take 592 seconds. Let this one take about 550 seconds, and the 18 one about 516, and the 20th about 476. Now the ninth step says label a test tube U and put 1 cm cube of U into test tube. Repeat 5 to 7 step using the test tube labeled U. So we are going to be performing the same experiment with the U to record its time. So let's suppose it takes about 450 seconds. Okay, so estimate the percentage concentration of glucose in U. So 450 will be less than the 20th, so it means its concentration is going to be higher than 20%. Let us suppose about 21%. Now next part says describe how you would modify this procedure to give a accurate estimate of u. So this is the how I am going to be modifying and bettering the experiment to have a more precise estimate of it. So first of all, I'm going to say that repeat the experiment with each concentration and take average time. The other point that could be is that use a colorimeter to reduce endpoint, and the other that could be is use the concentrations beyond and below the 12 and 20% concentration. Use more concentration above and beyond 20 and 12%. Okay, so this will be the improvements. So state two significant sources of error when carrying out step 7. So what could be step 7? Let me just check. Step 7 is measure time taken for each test tube to reach the endpoint. So the significant source of error in this will be that it is difficult to start the experiment for each concentration at same time, and the other could be difficult to determine the endpoint. Describe how would you modify this procedure to reduce one source of error as you stated in a part 6. So what we could do is that we could use a colorimeter. So I've already mentioned that in the previous part, but in this part what am I going to be answering is that time for each concentration separately. So now the B part says an investigation was carried out into the effect of drinking an energy drink on the blood glucose concentration over a period of 3 hours. So the results are shown. So this has both numerical values on the both columns. So plot a graph of the data in table 1.3 on the grid in figure 1.1. Use a sharp pencil for drawing graphs. So the range for the time is 3, and for the glucose concentration it is about 9.1, and the lowest is about 5.2. So let's check how many boxes we are provided on the x-axis. So it is 1, 2, 3, 4, 5 and 6. So we could actually use zero point 0.5 as a suitable scale. 0.5, then 1.0, then 1.5, then 2.0, then 2.5 and then 3.0. So it was time, and it was the unit was in hours, and the other axis it was glucose concentration millimole dm cube. So glucose concentration in millimole dm cube. Let's check how many boxes do we have. So it is 1, 2, 3, 4, 5. So let's divide the highest value, which was, let's have a we can also use a break to break the graph. Like we can start from 5 and we can then use the highest value as 10. So 5 divided by 5, we could use 1 as a single, you can say, as a single unit on a one box. So this will be the break here. We could use this break. So then we would actually have 1, 2 and 3 and 4 and 5 boxes. So let's just not use this break. Let's just try to handle it like that. So we have the 9.1 is the highest value. Let it divide by 5. So we could just use 2. Okay, 2, 4, 6, 8 and 10. Now let's check the first value. It is 0.0 and 5.2, 0.0 and 5.2. So 5.2 will be about like here. This will be the first point. Let's check the other. It is 0.5 and 7.5. 7.5 will be about between the 7 and the 8. So this will be the other point. Then we have 1 and 9.1. 9.1 will be about like here, not here. Let me just erase this point. It will be here. Here is the 9, so it will be midway between the small box. This is the point of 1 and 9.1. Then 2 and 8.4. So 2 and 8.4. So 8 is about here, and 2 boxes above 8 is 2. So this is the point. And then we have 3 and 7.9. So 7.9 will be about this point. Let's join it with straight lines. So this point, then this point, then this point and this point. Okay, this will be the graph. So this was the last question. So many of you are actually asking me that to attempt this paper. So I have attempted the whole paper. If you have any queries, you can ask me in the comments down below, and subscribe this channel, share it with anyone who you think it will help. Good luck for the paper tomorrow, and I'll be meeting you in another video surely about the chemistry practical. So till then, goodbye, stay safe and good luck.